Save It for the Blind is sponsored by Tetra Hearing. Tetra Hearing isn't just hearing protection, it's hearing perfection. Kershaw Knives. Kershaw manufactures next-level pocket knives for outdoor enthusiasts. Save It for the Blind is now also being sponsored by Roseville Motorsports. Roseville Motorsports is the greater Sacramento area's premier destination for all your power sports vehicle needs. Stop in today to browse off-road vehicles from leading manufacturers like Polaris, Yamaha, and more. Rig 'em right, heavily seasoned gear for the die-hard waterfowl hunter. If you guys are enjoying the show, we'd love to hear from you and get your support. Please follow us on your favorite podcast platform. Give us a 5-star rating. Your reviews really help us grow and reach more people. Thank you for being a part of our community. Nice. Well, Phil, I'll introduce myself first. I'm Carson Odegaard. I'm the hunt program coordinator here at CWA and then one of the hosts of the Save for the Blind podcast. And Jeff, you can kind of introduce yourself real quick.
Jeff Smith, Director of Hunting Education Programs. So a little bit of everything from habitat management stuff to camps to college camps, all that good stuff.
We are happy to have you on. This is going to be an awesome discussion.
Yeah, absolutely. Super sorry about that last time.
No.
At a different time, and we were out.
You got caught up in field work, man.
birds.
Hey, the more important things take precedent. That's definitely more important than this.
That's good. I was that day I was up all night lighting, so I had like 2 hours of sleep, and then I was like, oh, good, I can go back and take a nap now.
It just all worked out for the best. Yeah, we filled that spot, so it wasn't a big deal.
If you dish just a second time, you're going to be dead to us. All
right? You get a free pass on the first one. We'll get going. Welcome back to another episode of Save It for the Blind, brought to you by Tetra Hearing and Kershaw Knives now. So not only do we protect our hearing, but we have the knives to process our game after. So big shout out to those two companies. Today we have one of our usual guests, Brian Huber, CWA's Wood Duck Program Coordinator, and we have a special guest, Phil Lavretsky with Duck DNA. So Phil, thanks for getting on and talking to us. We've got lots of questions. Everybody here at the table except for Jeff is in the biology degree field, so he has the duck experience, but
excluded, man.
We have the academic experience. So I've got lots of interesting questions that we'll get into, but kind of just start. Let's take it down your path and how I know you were at Davis for a little bit and how the DNA started, and we'll just go from there.
Yeah, let's hear
good.
the Phil in the bag days, to Dr. Phil, give us the whole
you want me to talk about that?
Yeah.
That's still a dream. So I grew up in California. Actually, I grew up in Los Angeles and started hunting ducks when I was probably 10 in Salton Sea at Worcester area. And then that's what I did for a long time. I went to UC Davis, as you mentioned, for my undergrad. I got my undergrad there, and I specialized in genetics and wildlife conservation. So I majored and minored in those two things. And I sort of married them because I like genetics mostly because of Jurassic Park. But like
you were going to bring stuff back?
I want, yeah. And I'm getting closer to that dream. Maybe not dinosaurs, but at least maybe a passenger pigeon. But I was taking genetic courses, and obviously they're talking about Drosophila and all sorts of stuff. And I mean, I like the concepts of genetics, but that stuff bored me. And so, wildlife was my passion really to talk about. I started in fisheries. I worked at a lab there at Davis working on Sacramento perch, as well as hybridization between red bands and rainbows in the McCloud River drainage there. And so kind of got my feet wet there. And I love fish, but I really like waterfowl a lot more. So I finished that out. I actually then went and worked for Minnesota DNR, catching birds. And then I met Brian here at Cal Waterfowl, catching pintail and mallards and everything else there for a season. So we did that. And then and that was right before I did my PhD at Wright State University in Ohio. And I went there because Jeff Peters, my PI there, had funding, loved ducks, let me do what I want, and away I went with that. Because of my abilities to obtain tissue and do all the genetics, I essentially became more or less a hired gun for USGS, U.S. Fish and Wildlife, the Smithsonian, and others. So I would go on collecting trips, and then I would process them for genetic analyses. And so I sort of married the wildlife and genetics world. And now here I run a wildlife genetics lab, and by here meaning the University of Texas, El Paso. And I've been here since 2016, growing the program. And as you alluded to, the whole duck DNA project that I'm sure we'll be talking about is like the latest attempt, was the first attempt to do a citizen science project with hunters, essentially asking all hunters to participate and be technicians all around this great nation during their hunting season. And so we're sort of further incorporating hunters into the science background, just like we did with banding and other things, where we're asking them, the part survey and all sorts of stuff, where we're asking them for that kind of data. And so far we're marrying that, and that's been super awesome. So I'm sure we'll talk about that. So that's a bit of that story. So now here I am in El Paso in the desert, chasing desert ducks and growing and our understanding of waterfowl worldwide.
So when you started getting into all this genetics, how did it originally start? I mean, were you looking at Mexican ducks, right? With the genetics of Mexican ducks and mallards originally, or
how did genetics generally start, or how did it
for you,
my waterfowl genetics
for
start?
you and your waterfowl genetics?
I remember distinctly when I started my PhD, I sat down with Jeff Peters and I said, what should I do? And he's like, well, people like mallards, and nobody's really resolved the tree of life of mallards. And I was like, yeah, that sounds great. And honestly, the first thing was like, all right, so figure out, all right, how many species of mallard-like ducks? All right, there's about 14 of them. And then started figuring out, getting data for all of 14 species. And then we built a phylogeny, figuring that whole thing out. And essentially away went with that. And Mexican ducks nobody was studying Mexican ducks. We found a whole bunch of interesting aspects. There wasn't a lot of tissue available. So I went down to Sonora and then later on into many of the states in central Mexico and collected. And so that's how I started dove into the Mexican duck world, because not a lot of people were studying them. They were a subspecies at the time, but all of our work has really culminated in AOU, or our ornithological community association, elevated them back to full species because of all the work we've been able to do. So that was that. But that obviously, initially, to be fair, before Mexican ducks, it was American black ducks that really took my attention, which then led into the whole mallard issue that is now my attention, and then so forth and so on. And we've got work in Hawaiian ducks still, and Laysan ducks. And we were just in Australia last November with folks out of UC Davis and collecting there on Pacific black ducks. I had projects in American Samoa with Pacific black ducks in New Zealand and so forth. And Ramsey Russell at GetDucks.com, he even helped out and got us a whole bunch of yellow-billed ducks and African black ducks out of Africa. So we're kind of building that whole repository with everybody's help.
Cool. So, I mean, we all like mallards. So what's the evolutionary breakdown of mallards there that you've put together?
So what we put together, right? So I don't remember if any of you remember waterfowl class in 2004, 5, 6, or whatever year we were in. But there were these two competing hypotheses of how mallards got to North America. One was that brown ducks came here, evolved into our brown ducks, which is American black duck, two types of mottled duck, Florida, Western Gulf Coast, and the Mexican duck. And then mallards came. And then the alternative hypothesis is that mallards came, and essentially all our brown ducks are mallards who lost the green head. And all of our work now really suggests that that's the hypothesis that was true. A lot of times, and this kind of evolved from the fact that we were, there's all these keys, right? The white wing bars and all these plumages that we associate with hybrids for black ducks and mottled ducks and Mexican ducks. And when we started applying genetics to it, where we said, okay, U.S. Fish and Wildlife, call them as you see them. What we found was, in black ducks, they were only 60% right calling hybrids, right? Only 80% right on calling black ducks or mallards. 20% of those, each one of those should have been hybrids. With mottled ducks, 10% of the time, it was inaccurate that the white wing bars suggested a hybrid. And then with Mexican ducks, it was entirely wrong. What we found in each one of those cases is that when we saw hybrid traits, they weren't necessarily speaking that they were hybrids. in every single one of those cases, formative or hatchier males show mallard traits. And if they live to their second year, they turn brown. So we think it's a controlling mechanism. They all still have that kind of mallard genetics, but like, it's like being, 13-year-old boy, like you can't control your testosterone. It's just like all over the place. So sometimes those genes do get expressed, but they're splotchy, right? And it depends on what mom and dad gave you also and how that resolves. But yeah, if you get into your second year, you turn brown. If you somehow survive to about 6 or 7, you start showing those traits again. So we think, again, it's that testosterone, estrogen kind of control. Once you become sort of like an adult, you can control those steroids that are controlling the genes and basically down regulating all the mallard genes and up regulating that those brown duck genes.
So what's the general like timeline on the mallard kind of evolution and when they started kind of forming these?
Yeah, so now we've got
species?
several lines of evidence, but it's pointing to about 500,000 years ago, about 500,000 years ago, Eurasian mallard came over. And then
is that what you refer to as the old world mallard? When you talk about
old world
I've heard
mallard
you
came
talk
over.
about that genetics.
They stopped moving. The maternal lineages were about in the early 2000s. We estimated 500,000 years, and then now we have nuclear data, so the mom and dad part, and they also say almost every time when we independently test mallard versus a black duck or mallard versus a Mexican duck or a mottled duck, it's almost within that exact 500,000 time frame, where essentially there were a bunch of mallards that probably invaded, colonized. There's like all of North America, you can imagine, as a bunch of greenheads. And then essentially the glaciers cut those greenhead populations up into these pockets. And we know Florida and the Chihuahuan Desert around here were refugia for a whole bunch of species during that time. So
yeah, there was the Great Ice Wall or whatever, right?
Exactly.
Yeah, yep.
Yeah, so if you look at it, it would have pushed a bunch of mallards to this area where it was wet at the time. And then in the east, that would have been its own population. And obviously, as that receded, the eastern area became more boreal, darker. So there was that natural selective regime on both males and females to not get eaten. And then here, essentially, it was a bunch of mallards that no longer migrated, stayed. And because, again, both for black ducks and for mottled ducks and Mexican ducks, there aren't other ducks here when they breed, right, of that size. So picking incorrectly or sort of mating wasn't a true issue. It was really about just surviving. And thus they lost that dichromatism, I guess you could say, which is losing that need to put all those resources to look all fancy and stuff like a mallard does. And that, so far, all of the evidence is suggesting that that's how all of these species sort of almost probably simultaneously came to be, where these mallards got cut off 500,000 years. They came 500,000 years ago, and during the glacier periods during that time kind of cut those populations off, and they took, and they evolved to those, adapted to those regions and those ecologies.
So with the duck DNA research that's going on now, what's the importance of what's actually going on? Because I know people, I shot a hybrid, this and that, and as a citizen that does that, there's nothing more than a trophy on the wall. But what's the effect? Are we gonna breed out pure populations of ducks? Or I know I've heard, and this may be a completely different thing, the rumors of the farm ducks that are getting let go, mallards in the South Carolinas and that stuff that are interbreeding. So what's like the worry there, and what are we trying to do with this information?
Well, this is kind of this was like secondary, right? You kind of stumbled upon this when you were doing other stuff,
like
right?
The South Carolina mallard stuff,
right? How did the game farm bird issue come to your attention first? Because I know when you first originally kind of discovered it, there was a lot of pushback and a lot of, the first NADS that we went to 8 years ago or whatever. Everybody's like, who is this crazy kid talking about genetic
info?
Game farm mallards. Who is this kid? And now, at the last NADS, it was one of the main topics, right? So it's really come a long way. And I think if I remember right, the story you kind of stumbled upon the game farm mallard issue, and they just brought it to your attention. So
that's exactly it. Yeah. So the original story I was looking at was black ducks. So it's hybridization with black ducks, right? We had age-old question: are black ducks being basically bred out, competed out? Is that's what's occurred? And so in 19, I published a paper that I had been sampling since 2010, and this was all the U.S. Fish and Wildlife, all the winged birds and everything. And what was weird is that when I was looking at it genetically, so basically doing 23andMe, eastern birds, so mallards east of the Mississippi River, had this other genetic signature that kept popping up, and especially in the Atlantic flyway. And so that every time I ran any analysis, all of my analyses said I had three populations, or three genetic populations: black duck, western mallards, and this eastern mallard. So I had these three populations. So that was 2019. So then obviously I knew the history of North American love of game farm mallards. So if your listeners don't know what a game farm mallard is, it's simply a moniker for a type of domestic duck. It's like a khaki Campbell or call duck, or really it's like calling it more or less a game farm mallard would be a husky to a dog, right? So the dog is the wild mallard. It was a Eurasian strain. And mallards was one of the last successful domestication events by society, starting with the Ming Dynasty, and then Europeans had all their sorts of domestication events on top of it, whether you're doing it for egg production or meat production, fanciness, just like our dogs, right? There's all sorts of breeds. And this breed, as far as I can tell, started around the 1650s for the specific purpose of hunting, and by King Charles II, bringing wild mallard eggs in for captive breeding. And the first time the word, at least as far as I can tell, the word game farm mallard was written down were by the British in 1890s when they were banning them and releasing them. Anyways, fast forward to about 1910, 1920, really, right? North America decimated their wildlife populations. All sorts of work was started. I mean, the same kind of situation with turkeys where they were catching them, captive breeding, trying to move them all around. Well, in the East, they wanted mallards. And the history goes like they tried to catch mallards and black ducks and breed them in captivity. But of course, they're wild, so they weren't really producing very well. And so somebody must have known that the Europeans essentially perfected this breed that looks like a mallard, acts like a mallard, and puts out a whole bunch of eggs in captivity. And they brought them here. And since 1920, we've been basically breeding and releasing the same strain across North America. From about 1920 to 1960, it was roughly 500,000 of these things being released a year, largely on the Atlantic seaboard. And then since the 1960s, the best estimate is about 250,000 a year, with about 210,000 of those in the eastern seaboard. But I can assure you that I've spoken with folks that raise these things that state that even they themselves can do 200,000 by themselves. So I'm pretty sure that number is underestimated. Yeah. And the idea is that these things basically died, right? They had no effect on nature. You could just release them, and they were trained, as people say, to go to their ponds, and you could shoot them. And so they had survival is almost zero. And if they even left the compound of their wetlands, they died. And if they didn't die, they definitely didn't breed. So this was the dogma, and if that was the case, then they should have no kids, and if they have no kids, there should be none of their genetics on the landscape, and that was clearly not true. And as Brian alluded to, we went to the historical data set at the time because I knew I had to confirm the origins of those mallards, being like, well, maybe Atlantic flyway mallards were always different, but we confirmed that alternative signature is simply game farm mallard. In 23, we also confirmed that genetic signature is identical to what is in Europe, so Sweden and France. We got in England, we got birds out of there, game farm mallards, and the same exact thing. And so what we also confirmed in 23 is that on average, again, every state is a little bit different. If you're closer to Canada, you have more wild. If you're closer to New Jersey, you have more feral. There's about a 2% chance that if you're somewhere in the Atlantic Flyway, that you're looking at a true wild mallard and not a mutt or just simply a feral game farm mallard.
So you said only 2%.
Yeah.
2%, and that was down from 8% when we looked at it in 2019.
Oh my
God! In 2019, samples were from 2010, so technically that was 10 years. We went from 8 to 2%.
So everybody likes mallards, and it sounds like these states are putting more mallards out into the population. So what's the problem? Why is that a bad thing, Phil?
That's exactly what everybody keeps asking.
Yeah, what are these birds?
We're shooting mallards.
What's
who cares?
Yeah, what's different about
greenhead is a greenhead is a greenhead, and that's like beyond false. So I knew that question, right? Everybody would say, well, hybridization, sometimes it's actually good and sometimes it's actually bad. It's not. It's okay. Like a mallard's a mallard. So I partnered with Ben Luganen and in the Great Lakes area and all his partners there, and as well as Doug Osborne and Brad Cohen out of Arkansas and Tennessee Tech, to get birds in the Mississippi. There's political reasons, or I actually don't really know the reasons why we didn't partner with the Atlantic Flyway study that's putting out 1,000 or whatever telemetry units. But regardless, we got the Mississippi to ask that question. What is their migratory capacity? What is their breeding capacity? What is their behavioral capacity? Is there any sort of genetic linkage? And if so, is it positive, negative, or neutral? Right? And those were the kinds of questions that we wanted to ask, just like you did. In a nutshell, it'd be like if I was like, I really want to have a wolf population, but I'm just going to put a bunch of huskies out there because it looks like a wolf. And hopefully it'll work. And that's just not the case. It's not true. So what do we know? So in fact, 2 days ago, we have a JWM paper that got accepted, and then yesterday we got an Eco-Evo paper that got accepted. And both of those are showing a clear genetic association with migration. So more or less, essentially up to about 75% wild, it's almost a perfect correlation with your migratory tendency or lack thereof. Essentially, you have longer stopovers, more frequent and longer stopovers during your migration. If you move, you have a significant increase in your urban capacity. So you're moving between urban sites. And this was especially astounding for our Great Lakes birds. If they left, their migration was effectively a random walk. They did it, and they hit Tennessee and basically went back or died. And effectively they've lost the capacity to either queue and or be able to move, have the fuel and the capacity to do the migration. And where that comes from is we brought these birds into captivity. Hopefully these couple papers will be out here soon. And essentially what we're seeing is that this will be published. They have a 50%, with females actually showing 75% reduction. So a game farm male and a game farm female compared to their wild contemporaries, reduction in feeding and being able to feed on wild seed. So that means they have to feed twice as long and twice as much wild seed to get the same calorie count.
And that comes from the selection of humans that selected the game farm birds to pick up food in a pen, right? So they have this adaptation.
So we've associated that with changes in their bill. So game farm mallards have shorter and fatter bills. So their lamella spacing has completely been changed. Yeah, I mean, like Brian here alluded to, they also have longer legs and shorter wings. So you can imagine the king's man, throwing out bread and corn. The thing that gets bigger and gets to breed was the one that ran over to the corn and the bread versus waddle over. So you do that and you chop off the head of all the ones that waddle versus run over and be able to peck. Well, it's pretty quick that you can turn a chicken or a duck into a chicken, right? And that's our data shows that that's basically what we did. But anecdotally currently, we're literally using a DEXA scanner to look at fat content. But these birds were put on Purina chow. Wild birds, after our feeding trials, went straight back to 1,200. Your 900 to 1,000 for female, 1,000 to 1,200 gram bird. These things like cannot go back to 900, like past 900 grams. And it seems like they have physiologically, they've been changed to basically not put fat on, because they're the same length, but they're like 300 grams lighter no matter what. Right. So the only way to do that is you lose fat or you lose muscle. And so now so that was independent of all the telemetry data that we got. So again, about 700-800 birds with telemetry units. And these birds are basically these shortened wings. So you took a jumbo jet, you made it into a fighter, but you essentially took its fuselages off, right? So it's flightier, but those flight patterns can only be sustained for a short period, which is all people really wanted, right? You're supposed to throw them out. They like come in like a little widgeon, and you get a sportier shoot at that moment, right? So that's more or less what we got. And people have told me like, oh, my birds are flightier, and I never understood what that meant until now. I was like, yeah, you made them flightier for like a very, very short period of time. And so that totally makes sense why their stopovers are now so frequent, right? You've got no fuel, and you're probably pushed to move because of weather, but you can't, right? You have to stop. And the reason for that urban stop is because that's probably where they're getting any sort of calories to even get to the next stop. And that's why their movement patterns, hypothesis still, are random, because they're just basically looking for their next meal. It's like getting off the freeway, eating McDonald's, getting back on the freeway, and you're hungry in 2 miles again. You're just like, where's the next McDonald's? It doesn't really matter where it is.
Yeah.
So that's the first thing, is that, so to answer it, no, now we have a genetic, behavioral, ecological, and generally biological link that is distinctly different. And what's more important is that when we look at the hybrids, they essentially independently, we find this break where once they get to about 75% or higher wild, they look wild, right? They act wild. They are wild. So that's about 2 generations of backcrossing. That does mean that natural selection is constantly like killing off a bunch of birds out there, because they're not flying, they're not moving correctly, they're not eating, right? All that time that you have to spend eating, that's increased predation risk. So all those birds, even if they wanted to get away from predators, again, that's probably the urban setting, right? Not a ton of aerial predators and other predators where they can survive on parks and whatnot. But these birds are probably getting hammered as well on it. So survival rates are down. But the kicker, also going to be published soon with Ben Luganin's work, is that they were able to go and identify, right? So they were all about 600 nests or hens. And so for every hen, they could go and see, did the hen sit on the nest, finish the nest, leave the nest, get predated, and all this stuff. And more or less from that, what we found is that you need 3.3 females of basically female mutts to equal the production of 1 wild female mallard. So it's a 3-to-1 ratio.
Horrible mothers.
And more or less, his text basically, they start making a nest. They usually get off the nest in only a week or 2. And when the text went there, it wasn't predation. There were literally eggs just kind of strewn about, right? Just like a chicken would do. Because that's what more or less they have no innate sense to sit on a net, to make a nest and sit on a nest. And we saw that in captivity as well. A wild bird will make a nest, but no eggs because she's all stressed out. But these things, no nest, or like half-ass make of a nest, but there's just eggs everywhere. You can go in there, as long as you're giving them Purina chow, they'll still make an egg.
But you mentioned the wild mallards, it's like 2% being harvested, and it was, 8 or 10% 8 to 10 years ago. Why the drastic decline? Is there just more game farm mallards on the landscape? Are they competing? What's the deal there?
We will be publishing this. So we went back to the historical samples. Thankfully, there was like, people loved hunting ducks, and they put them in all of our museums, right? So we partnered with 27 national museums, including the Smithsonian, and we got mallards from 1800 to today to ask that question, because the only way for that to happen is you have no more wild pool, and all you have are hybrid combinations that basically swamp everything out.
Okay.
And what we found is that by 1970, before 1970, there are these specific ebbs and flows in the Atlantic, the Mississippi, none in the Central, but there was one little like pop up in the Pacific Flyway in the 1920s of game farm ancestry being on the landscape at significant levels. Then essentially the Central and the Pacific went to zero, right? Natural selection parsed all that stuff out. Big wild mallard population, back crossing of 2 or 3 generations, and your great grandkids are wild, right? But what happened in the Atlantic Flyway is that signal never went back to zero come 1970. So since 1970, it's been just bumping up and down, just ebbs and flows, but it never came down, right? Even the Mississippi Flyway, it showed a similar trend as it bumped up in the 50s, 60s, and 70s. But actually in the 80s, that thing went straight down to zero again, and only recently has started to spike out. So that suggests that they stopped, maybe during the Dust Bowl and all that era, like people are like, okay, let's get production up. And then years of good production in the prairie potholes and whatnot. Maybe the Mississippi's like, no, we don't need these. But the Atlantic Flyway always have been, that's been part of their culture, is what, folks have said, because they've been doing it the longest. So it's just natural to be like, all right, let's put out some birds out here and shoot those. But more or less, they've lost the wild signature on the U.S. side in particular, of wild birds even breeding on the landscape. So I'll tell you a couple states: Massachusetts, Connecticut, New Jersey. When I look at just summer birds, so these are your what you expect to be your local birds, I cannot find a mallard that doesn't have substantial amounts of game farm in it. So I can't find just a wild mallard that is being banded in any of those states for the last decade.
Are these game farm mallards still being released? And if so, is it by like the DNRs? Is it like private hunt clubs? I know we brought up South Carolina before, and we've talked to some folks that's essentially what they do. But is that just a thing of all these other states? Like that's still a normal practice?
It's still a normal So it used to be state and federal funds were, because again, back in the 1920s, right, everything was decimated. So they were trying to bring back everything through these kinds of actions. We even know in turkeys, right, if you use a domestic turkey, that thing doesn't take anyways. Or at least we learned from that. And so, but that, like I said, it's more cultural. So the state and the feds, at least as far as I know, don't participate in it. There aren't any tax dollars, at least I hope not, going towards it. So it is all privatized, right? So that's, in my opinion, a little worse, right? So it's a private good now polluting a public good, which is oftentimes my problem. If they truly just died there, I guess, like, if you want to shoot chickens, I guess you could do that. But it's having a serious impact on the viability, migratory capacity, general viability, and fecundity of our wild mallards. And it is unfortunately ticking west.
Right. So
like
that
the prairie pothole region is large and it can sustain, but obviously, anything's possible, right? There were billions of passenger pigeons and hundreds of millions of bison, and here we are.
So the big concern about the game farm mallards is it, like you're saying, spreading west, kind of starting to infiltrate like the prairie potholes region of our migratory birds, and kind of having that same influence, the Atlantic Flyway, where they're just not as viable as the wild mallards.
Right. I mean, if you wanted to have concern, we have increasing amounts of data that at least partially explains why you don't have, your Halloween mallards in the southern part of the Mississippi Alluvial Valley, or even that connectivity to the southeastern part of the Atlantic Flyway, North Carolina, South Carolina, Georgia areas. That was largely fed, that secondary push into the MAV, and then primarily pushing into the southeastern area of the Atlantic Flyway were those Great Lake birds, right? Especially out of Michigan and Minnesota. At least that's what the bands show. And if that population which we now know has been completely transformed into more of a random walk that don't like to go south of Tennessee. Well, that sure as hell explains like birds no longer coming down, right? You could have all the best weather in the world for birds to be pushed, but if they can't queue and can't get food and process it properly, when unless we've got, I guess, corn or golf fields everywhere in the prairie potholes at that point, they're not going to come down. They can't. They, even if they wanted to, it doesn't seem like they're hybrids and they themselves have the capacity to.
Yeah. So how's the Pacific Flyway looking still? Are we still holding strong
we're
with
the
pure. Old world mallards, the Western mallard?
So Brian, I'm going to be able to tell. I wish I analyzed all the data out of the
that we just sent you?
Yeah, that you just sent. But that actually just got here. We're finishing out the last sample, so I will be able to tell you in about a month. But the samples I got when we did the egg salvage.
Yeah.
I think, don't quote me, but it was 10% or so were game farm mallards. And I know that there's I'm not going to name anybody, but there is one substantial producer in California. In fact, if you go back through our texts years ago, probably in 18 or 17 or 19, something like that, I was like, because I had a handful of mallards from Davis, right? Like 20 or so mallards all across the state or whatever. And I was like, there's no way there should be any game farm mallards there. And then I found it and I texted you. I was like, what the hell?
Yeah.
So they're there.
Is that where we?
To what extent I don't have the numbers yet.
The Gmail bands.
Is that where your wife went undercover to get some samples for you?
No, that was going to Jersey. Yeah, no. And so.
Yeah, it'll be interesting to see those new samples we got you. If that 10%, if that's similar or not. I mean, did you? We'll get into the duck DNA part later, but did the duck DNA show any kind of increase or anything on the Pacific Flyway?
We don't. I can't tell you. I don't have enough from those states, right? So we were too late for Alaska. We only had a few out of Washington, Oregon, and California that made it into duck DNA. So I can't tell you a whole bunch. But what I can tell you is that the trends were similar to mine, where east to west it declined. I think I found only wild mallards out of and there were a couple of special cases of Mexican duck out of California. Yeah, so that's its own little story. But it'll be interesting with this more intense sampling to figure out like, what's occurring in your kind of breeding pop in the Klamath at least, and that should be a pretty good tell.
So back to the game farm stuff. I mean, was it France that had they were releasing like 1 million? I mean, I can't remember the guy's name, but he was at NADS, and he was NADS. We talk about as North American Duck Symposium. So when we reference that, it's talking about ducks. But there's crazy story from other country where they basically have wiped out their entire population, right?
Yeah, so in that 23 paper, I knew I was going to have to go to Europe to see like maybe there's like what's happening there versus what's happening here. Because like I said, in Europe, they've been more or less doing this for about 400 years. We've been doing it for about 100 years. And you could just see it, right? Like France is this like epicenter, kind of like Jersey's our epicenter. And like as you go away from it, it becomes more wild. More or less, France is releasing somewhere actually between 2 and 3 million mallards right before the hunting season, so that way, quote unquote, they have a fall flight. And so they're like 200 years ahead, and it's stocking, right? It's kind of like salmon, right? You start stocking and it gets messed up, and then you're like, well, I got to put more salmon out,
right,
so you can get a run, and then it gets more messed up, and you're like, well, I guess we're going to put even more salmon out. And you know that if you don't put salmon out, you're going to have close seasons, because there's just not a ton of them that have the innate response. And potentially you could get those runs back, but you'd basically have to close that season for 10 years, right?
Yeah.
Something like that. And the fear, I guess it's my personal view, is that at least I don't know how it would look with mallards. It would be unfortunate that a species that is nowhere near the endangered species limits of millions of individuals and all this stuff, great habitat compared to songbirds and other organisms that are declining, yet we're still screwing it up, you know? it seems like an unfortunate fact. Like, can you imagine if like our stamp dollars went to go buy mallards so that way
release right before duck season
to screw up the genetics of the wild population?
Just like with pheasants, right? In Ohio, I knew that. All right, go buy those pheasants. I'll be there the next day after the youth could hunt. So that way, I shot a couple pheasants in Ohio. Ohio used to have pheasants. Then they died. They started stalking. You can't stop stalking once you sort of start, because the individuals in a cage are never optimum for the wild, right? They're always going to be. Everybody wins in a cage, but not everybody wins in wild, is what I basically say, and that means you're just pumping individuals that are going to breed to some extent and are going to then decrease the viability of your wild pool through time. And I was going to say this. So I had somebody come to me and say, well, hybridization should increase diversity. And I was like, yes, if it was like wild, right? But this is a domestic strain that has almost about half the genetic diversity of a wild strain. And so then what I did was I immediately had my former PhD student look at our full genomes, our whole genomes, and ask the question, what is the inbreeding coefficient of an individual or a particular ancestry? And it was almost a perfect correlation. As you had more game farm in you, you were more inbred. So that means a pairing between a game farm and a wild, right, is creating a nest that it has twice the amount of inbreeding than a wild pairing. And again, that has, and we already know that that's a case in quail, and it's a case for trout and salmon. I mean, fisheries have known this for a while, but this is so the interbreeding between a group, an organism that is highly outbred, lots of diversity, with something that is quite inbred, it does indeed inbreed the population, and that's what we're seeing.
Yeah.
Which is unfortunate.
I mean, you kind of alluded to it, but it seems like if we were just to completely stop planting these game farm birds over a couple generations, we could potentially kind of manage that genetics, right? Get that out of the system. Is that kind of...
Yeah, in theory, that's how it would work. Now, it would definitely, I think, work for the Great Lakes better than the Atlantic, because you've got that prairie pothole population that could hopefully naturally sort of come back into that area. But the Atlantic flyway, I don't know. Yeah. I think that would require putting wild individuals to try to basically reinvigorate the wild gene pool, and that start cutting out game farm and putting back wild. Because like I said, given that those summer birds, and it's very specific. So summer birds in Massachusetts versus summer birds in Connecticut have a very specific ancestry where it's like 80% wild, 20% game farmer, 70 and 30, or whatever it is. And if you could take a whole bunch of random individuals and see exactly the same ancestry and it's specific to those locations, that means those are just hybrid swarms, right? An 80-20 mom and an 80-20 dad is always going to make an 80-20 kid.
Yeah.
That means there's nothing to change. They've got no change that can occur without some form of input.
So I know there would be a ton of red tape and permits and all that, but potentially you could, is it possible to use wild genetics, right? Like kind of how we use our egg salvage program. We have wild birds. We put them in an incubator. I know they don't produce as much eggs and all that, but would that potentially, I mean, could you get it to scale where you could actually be releasing wild birds instead of these game farm birds? I mean, is that a solution?
So that was the email I was going to send you in like 3 days. Yeah. So we published and built a model to ask that question. We built it for Hawaiian ducks because a very similar issue with much smaller scale, and we're actually attempting what you're asking, right? We built models that have a bunch of variables, but the main output is management. Conduct a management strategy, and your management strategies are supplementation and or removal, right? And you ask the question, what would you have to do to change the genetic signature of this population with starting ancestry of whatever, and how long would that take? And in theory, our models show yes, in only about 3 generations, which is about 3 years, you could do it. And in fact, they weren't meant for a population. They weren't really designed for like 500,000 birds. And we'll have to make a few changes there. But I did run those models for 500,000. or what we consider the Atlantic flyway. And technically, in 4 years of putting out 50,000 birds a year, you could change the entire pop to over 90% wild.
Wow.
Which is huge.
Yeah, that's a big
in
deal.
theory. Of course, Mother Nature could like totally be like, maybe those wild birds are just like, I'm going back to the prairie potholes.
Right.
They're all
But-
excel those birds, and they come back here.
Yeah, but that's why. So we're working with folks in South Carolina. In fact, a specific person out of Louisiana with Panola aviaries, where we're setting up the breeding experiment. In essence, we're gonna mimic what today's usual like game farm breeder looks like. Then we're gonna make another cage where it's more wild-like, and we'll kind of figure out and ask the question, what kind of production can we get wild mallards to produce, and if so, what would that look like? So currently, one female game farm on average makes 100 eggs, and of course that's 100 viable eggs, right? You're just picking them up, putting them in the incubator, so they're going to survive. Maybe 1% don't. I don't know, but I mean, you can imagine it's 100%. So you get 100 birds out of one. Currently, just anecdotally, when you put those wild birds into the same setting as a game farm, they don't breed. But if you put them into a more natural like environment, in fact, if you put like a pintail in there, they'll nest, right? So the question becomes, how large of an enclosure would you need with the right settings to get them breeding, and how many nesting events could you get, right? They're still wild,
Right?
So my consideration would be, I think we could get 2, potentially 3 on a good caloric diet, but each one is going to be capped at 9 or 10 eggs. So you're looking at about 20 to 30 eggs compared to 100 eggs.
Right.
So now you're going to have to figure out, okay, well, how does that look? And of course, you're going to have to also think about how many times do we have to take males out of the wild to just reinvigorate, make sure we don't start seeing creep in inbreeding and other things that happen, and basically do the same thing as the salmon people do, where you have to bring in a few new salmon in, so that way you kind of reinvigorate it. And all our models have all of that incorporated in there, so we could technically test it. But now we just need some of those baseline data, like so production is now 30 eggs, right? So if we do a hen for every 30 eggs, 100% survival. What does that look like? How long does it take to grow 50,000 birds? Some of these guys tell me, they're like, oh yeah, I could get you 250,000 birds tomorrow. And I was like, well, yeah, I mean, those birds are designed to do that. They were developed by us to do that. And that's just not going to be the case for wild. But there's no way in the world that not only hunters, including myself, but the government would ever let me go and take 50,000 eggs, right?
Right.
So now, so really it's production. So can we produce wild, and what would that look like?
Right.
And that's where the California egg salvage, and that's where my email is going to be like, Brian, how many eggs do you think you can get me? And my hope is, if I work with California and a few folks out of South Dakota and North Dakota, including if there's any hen houses out where we won't take all the eggs, maybe we'll take 50% per hen or whatever. Maybe if we can get a couple hundred off of all of that, and Minnesota maybe could help out. Yeah. We have enough to at least pilot it and figure it out. And then once you establish it, I think we could then move it to these other breeders if they're willing. And in essence, they would do what we need to do, which is start pumping out wild birds, right?
Yep. Yeah, exactly.
And so, because currently they're doing the exact opposite. It won't work if like 50% of the breeders do that and 50% of the breeders still do game farm.
Yeah.
Because then essentially that's just, you're negating what you're trying. And in that sense, we just have to keep talking, I guess.
Yeah, I mean, at what point?
And laws would be.
Yeah, exactly. At what point do we have to does Fish and Wildlife or somebody have to step in and say, okay, this isn't good for our wild populations. We have to form some kind of regulations.
That's a great question.
Right?
I don't have an answer to that.
Yeah.
So, I mean, that's more societal, I guess, talk, because there's 2 problems there. So that in the rules, a game farm mallard or any domestic is not protected under Migratory Bird Treaty Act, right? That's why you can breed, move them, sell them, do whatever you want. You in New Jersey, you can hunt, you can shoot as many as you want between 10 and 2, because apparently wild birds don't fly between 10 and 2.
Yeah.
Before 10 a.m. and after 2 p.m., you have to abide by whatever the bag limits are. But between then, you can shoot 100 mallards if you want.
That's crazy.
That's wild. So I feel like people are going to start having DNA tests in their kids. Be like, this one's farm. Like, here's your proof. Like,
right. Yeah.
Well,
yeah.
Fun fact.
yeah.
So things that like the data set that Brian's sending us over, the data sets that we're building with duck DNA that we'll get into, those are the data sets we're now all the pictures we're pushing into an application we're trying to develop. We've got the brain, we just need to get enough data, but more or less it's looking like currently it's built for model ducks, Mexican ducks, and mallards. But basically, what we figured out, because there's morphological, those changes in bill and tarsus and wing, if you could get a particular picture where it could scale them, those ratios, we can almost 100%, well, 98% discriminate between just a wild and a game farm.
That's cool.
We're over 90% accurate for those super early hybrids. So imagine if we can get this application first into banding crews, so we can start separating these things out for survival estimates and all that when we're banding, and standardize the whole damn thing. But in hunter's hand, technically, if everybody got on board, and there's lots of people that would have to get on board, but yes, you could take a picture, and if it said feral mallard, maybe it doesn't count against the bag. Right?
Yeah, you're doing a good deed by taking it out of the population.
And more because the alternative is, so through, again, duck DNA and some of my state partnerships, we're trying to create fine scale maps of the state of basically looking for hotspots, right? Because technically, if you could find a hotspot, if you got wildlife refuge areas or places you could get hunters in, maybe there could be a draw, and essentially you're the wall, right? So if you get drawn, you get to shoot 10 mallards that day in that area.
Right.
More or less, in my mind, if we can't stop the release of them, then let's try to drive that survival rate to as close to zero as possible.
Yeah. So where do you, in 5 years from now, Phil, where do you see this whole game farm issue? Do you see it creeping just farther west? Do you see it kind of something happening? I mean, it's been fun watching your journey, because we've been friends since probably 2009, right? And we didn't know anything about game farm mallards back then. We were just trying to shoot rocket nets over pintail. But now, I mean, when you were at NADS in Canada, we were up there, and that was kind of your big breakthrough, right, with a lot of this stuff. And there was a lot of pushback within the community, and now, you're pretty vetted. And the new NADS, I mean, that was one of the major topics of the new the Duck Symposium.
It was mind-blowing to see that transition. I was walking around, and the first time I ever went to, at that time, NADS, I was probably the only genetics talk, right? Everybody's just like, oh God, what is this crap? This last one, it was like a lot.
It was heavy.
I was just looking through, I was like, oh wow, this is intense.
Yeah.
People are talking about like concepts like F1 and F2 hybrids and all that. And somebody turned to me and was just like, dude, this is you. This is because of you I have to listen to genetics.
Yep.
I was like, that's right. Now, what do I see I didn't see us to be as far along as we are right now. I mean, technically, we only started this in 19.
Yeah.
Right? It's only been 4 years, but we've learned a whole hell of a lot with lots of good partnerships. It all depends on the partnerships and social wants,
right,
more or less, right? The more of these kinds of conversations, the more people that are concerned, in particular, more people that have the right leverage. I mean, that's going to be end all be all, right? So
this kind of flows perfect into your partnership with Ducks Unlimited and other organizations, right, to start this whole Duck DNA program. So what's Duck DNA? How do you sign up? I've tried to sign up. I don't think I got selected yet, but luckily I know you, so I can just call you and hopefully get some samples.
We haven't picked the middle band.
Oh,
right?
gotcha. So I still have a chance.
It's about to be picked in a week or so.
Yeah.
We only picked the northern. So Duck DNA. So this is only year 2 of Duck DNA. So Duck DNA is a partnership between my lab here at the University of Texas El Paso and Ducks Unlimited with Dr. Mike Brazier. more or less, right? So I was seeing all these patterns, and in the back of my mind, I was like, man, if I could just get like these like a tube in hunters' hands, we can monitor this thing at the landscape level, right? We can do it across space and time, right? Every year we could look at game farm ancestry across all 4 flyways, going from north to south through the hunting season, basically tracking that progression and what happens. And so Mike, at the same time, sort of was thinking of it, given, I was on a couple of DU podcasts and I was on other podcasts, and this topic kept coming up. And he and I sat down and more or less like said, let's do this partnership where Ducks Unlimited is the face and the, reach, and then we'd be the lab, right? And so the first year, last year was our first year. Almost didn't get it off the ground because they're like, there's no money. And then 1 month before we're supposed to start, here it all is. And so we pulled it all together. Thankfully, we had 4,200 applicants, 49 states, everything besides Hawaii, all for 300 kits, right? That's how much funding we had. This year, we have 700 kits, 3 samples per kit to kind of increase. So last year was 5 samples. This year's 3 samples per kit, just to increase like how many people can, how many kits we can get out there in people's hands. So 700 kits, 200 just went out. Again, it was the northern part of the
how many applicants this year?
States. So we are currently, I believe, in the 2,500s or 3,000, close to that. So we're getting close to our 4,200. So I'm super excited about that.
Yeah.
I think that's the number. I haven't heard it in a 2-3 weeks, but we were at like 2,000 some at that point. So I assume somewhere around there.
Yeah.
So lots of interest, right? And it's been hugely popular, including with the fact that like we got several emails of folks being like, my kid didn't want to go unless they could do duck DNA. Can we have like, if they had one, they're like, this is the reason my kid went hunting, or could we get a tube? And they'll be like, of course. So obviously it's like an engagement tool.
Yeah.
It gives something back, right? If you shoot something, you get a certificate of ancestry, just like you would have a band return.
Yeah, we have one for the one that you guys sent me. We'll get into that in a second. But the process is extremely simple. You get a kit sent to you, everything's in there, and you basically cut a 1/4 inch off of the tongue, put it in the sample, and send it in the package that's already there for you, and away you go.
Yeah,
right?
we tried to simplify it. It used to be, well, it still is when I do it, like more complicated, trying to like really limit contamination, all this other stuff. And then I was like, okay, the only way this will work is so I went out and I shot birds, and what I did was I did it the complicated way. And then I did it the like
the hunter way.
I'm the hunter. Like, let's just pull the tongue out with my finger, cut it off, put it in a tube. Like maybe wipe the scissors off. Maybe
use the same scissors I use for all the species I shot.
Exactly.
Right, yeah.
And so then what we said, okay, so this is how much we need. There's a brand new buffer. Thankfully, it worked. And then what we do is we take the center part of the tongue. As much as I want to trust everybody, I can't. So we take the center part of the tongue and extract DNA. And last year we had close to 1,000 samples sent in, and I think only less than 1%. God, it was probably only like 10 samples we just couldn't get to work.
Yeah,
like that's pretty good.
Yeah, no, that's amazing.
For a bunch of samples that probably like were never really frozen, thrown in the truck, just like driven around.
Yep.
Like that's pretty. When people were like, well, what will be like the failure rate? Like I was like, I have no idea. But that was really great to see that we could get them to work.
Yeah, it's funny because in a lab
you're
so careful not to contaminate anything. You're sterilizing everything, and now you got duck hunters out there just whacking off tongues, throwing them in a bottle, and
and it's working, right?
Yeah.
One thing, the other thing that I was afraid of was like, are we just gonna get a bunch of DNA from the food,
right?
On the tongue or whatever? And I bet it's there. Maybe in the future we'll see if we can't get diet off of them. But yeah, no, it worked, and it's continuing to work right now.
So you guys
now we're basically using that method for the states, for the feds. If we're not bleeding birds, we're basically asking people just like forget the muscle, just do this, and it's been working.
So you got a year of data now, right? The pilot year is complete. So what did we learn from duck DNA? What was some highlights from it?
Yeah. So what did we learn? A big one that I learned was that in Florida, that game farm mallards are breeding with muscovies. That's cool. So there's been a few of those. We got some wood duck mallard hybrids. The general trend as far as the game farm thing is occurring is that it's still an east to west. We just don't have enough sampling to see how it compares to our thicker sampling. Hopefully this year with the 2,000 samples that we're sending out, we'll get a better estimate. But what I did learn, and something I should have thought about that I didn't, is a 3-species hybrid. So you have a mallard game farm, and they interbreed with a black duck, right? So you have a hybrid making a hybrid.
Right.
And I should have thought that that's possible given how many hybrids are out there. But this made it way more evident. Like I could see it a lot clearer. And that trend is still, Atlantic Flyway has a lot of those. Mississippi has a few of those, and so we're starting to see that.
Then identifying the female hybrids too, that was a big
and that was
thing.
the biggest thing, right? So one of the other aims of this is asking the question, well, so there's brewer's ducks and other mallard, basically mallard and whatever hybrid, right? The first question is it a wild mallard or a game farm mallard?
Right.
That's the mallard source. And the second part, is it always a male, right? Are females sterile, or they're not viable, or whatever, or is it because like we just can't identify female hybrids very well, so that's why we're underestimating hybridization between mallards and whatever, right?
Yeah.
And right, that last year was the first time we could genetically vet that female brewer's ducks exist, right?
Yeah.
And that so we always also thought that those hybrids would be inviable, but you got us a second generation hybrid. That means a hybrid brewer was able to survive and breed,
I don't know
right?
if you can pull that picture up, but a friend of ours, Josh Gibbons, shot this duck, and he sent me a picture, and he goes, "Brian, what the heck is this thing?" And I said, "Oh, that looks like a brewer's duck." And man, I got so much crap for saying that. I put it on Instagram, and everyone's like, "You don't know what you're talking about, blah, blah, blah." Even some colleagues that we're really good friends with, disagreed with my assessment. I was like, "Hey, maybe I'm wrong, but I know how we can get to the bottom of this." So I called Phil, and I was like, "Hey, can you send me one of those kits?" So sure enough, I go to Josh's house. I dig in his freezer. We cut out a piece of tongue. Send it in, and sure enough, if you can bring up the certificate. This is kind of the certificate that you get from duck DNA. It's pretty impressive. It should have Josh's name on there, not mine, but we haven't got that squared away yet. But as you can see, I mean, it's amazing. It shows 76% gadwall, 24% wild mallard, and then it also shows the parental lineage, right? It was I can't read it from here.
So that male carries mallard, the mallard female lineage, right?
Yeah.
So that means either grandma or mom was mallard, because mitochondrial passed to all kids, right? So whatever mom is, everybody has the same mitochondrial. Dad doesn't provide anything as far as the mitochondrial lineage goes. So that's how we can parse that out. I was going to tell you also that with the genetics, we can identify sex. And I will tell you that as hunters, we're generally still bad at identifying black ducks, largely thinking that they're female mallards all the time. And every time I'm like, no, the genetics says male. And then when we would get a picture like that, I'd be like, no, this is a male. So, we got to figure that part out somehow one day.
Yeah.
But so this bird, we should really publish on it. But so hopefully after this year, we're going to do like a pilot. We're going to do a publication of like this citizen science, hunter scientist, this venue that nobody else has tried, and showcasing some of the things that we have now learned. So we've learned that those hybrids are viable to some extent.
Right.
We've learned now that we've identified female brewers and male brewers, right? We have a couple of mallard pintails. We have a couple of mallard widgeons. So we know that that's occurring. And one of the questions that I had once I started seeing this is, could there be like a way to say like, oh, this year there's going to be a bunch of brewer's ducks because a bunch
in
of
this
mallards,
area, right?
A bunch of mallards settled, a bunch of unpaired drakes settled where all the gadwall are, or where all the pintail are, or because so some of our other colleagues are showing this huge disparity in male female sex ratios in mallards. So it's like, where are all those males going? Right. And I believe, I want to say, I think that the majority of these kinds of events are all during secondary nesting through extra pair copulation. I don't think that like they're mate pairing on the wintering grounds. It's all this is all occurring
forced
on
copulation, we have to call
grounds.
it, right?
Yeah. And so like, could you almost be like, everybody gather up, you're gonna get a chance at some brewers this year, because they like all these males
shoot everything that's brown and maybe green.
The other interesting thing, and we don't have enough data for this, but this year there was Mississippi was a hotspot for brewers ducks. Again, that could be totally just like the fact that those are the people that sent them in or whatnot. But the other interesting, this one year, there was a study about trying to figure out like hybridization between all these different species by Siebert Rower and others, and it took them 10 years to get X number of birds, and we beat that number in 1 year. Right? All of us may kill one of these types of birds in our lifetime, but together every year we could get these birds and start asking those kinds of questions.
Yeah, and so with duck DNA, even if you don't get selected and you shoot like a really cool hybrid, they can still get in touch with you, send you a picture, and you could be like, all right, that one's worth sending a vial for. Let's figure out what's going on. So anybody that's listening,
we even had like people like, hey, this is my kid's first, and it's just a mallard, maybe it's a hybrid. Could we send it in? We're always just, I always have to ask, do you? But yes. 100%. Yeah. So we're always, if you kill a hybrid, we have a whole pool of tubes just for hybrid participants. So if you're out there and you shoot something super cool, throw it in the freezer, contact us, and we'll get you a tube, ASAP, and get you that info.
What if something's already mounted? I mean, are you guys able to pull DNA off anything on like taxidermy? I would assume not, but
the short answer is yes, it's possible. No, not with these methods. You'd have to send me maybe $500.
Yeah. I mean, that's how you did it with the Smithsonian samples, right? I mean, you guys got tissue, but obviously it's a more expensive process, but it is possible. Yeah.
It is. Yeah, exactly. So it's called ancient DNA protocols that we optimized. And that's exactly how we got, birds from 1800. I was up in, in Albuquerque at the museum there. And I was handling a bird, and I look over, and I'm like, "Oh my God, it's Aldo Leopold!" I was like, a bird that he shot and put it in. I was like, "Oh man, we're definitely doing that."
That's so cool.
Nice.
That's awesome.
So Aldo shot this bird, probably like starting to understand, maybe had ideas of what genetics. I mean, we barely knew what genetics was at that time, and like put it into museum, and it became a time capsule, right? And now we've extracted DNA from a bird that was shot 100 years ago. So that's the importance of these museums, is they're basically, you don't know what technology is in the future. So these things are super important.
Yep.
Like, how long does it take to find the DNA of one of the taxidermy birds opposed to one of the birds that someone sends you in a half a tongue? You might be getting a lot of $500 bills.
Jeff's ready to send you $500.
The methods are a lot more time consuming, and the stuff is a lot more sensitive, because you don't, it's forensics basically. You're dealing with very little amounts of DNA, so you got to be able to be very careful not to contaminate and all this other stuff. Usually we can do it in about 2 weeks to get the DNA out. Here, with your tongue, we essentially have DNA out and it already going through the process in about 36 hours, so day and a half. Yeah, and we're done with it in 3 days, and then essentially we have to wait for the core to sequence it and stuff. So we don't do that part in house because it's super expensive, and cores are better. So hopefully we don't hit holidays or whatever, but usually we can get that back in a week. And then analyses, I've basically optimized for almost just 1 day. So your bird, from us getting it to you getting results, the most optimum would be about 2 weeks. in theory, if you paid me a lot more, you could get it in a week.
If you sponsor,
but again,
yeah.
but again, it's like last year, it was about 2 or 3 months. It was more like a month to 2 months, and that's because we hit the holidays, and then we were at Ducks, and we got like, oh God, like 500 samples, and so we had to catch up on all that, and so that's why so hopefully this year, by going from 5 to 3, we can get more people submitting them quicker. Because I think it's like a limit, right? It's like, oh, I got one, I'm gonna wait, But if you only have 3, you shoot one, and you're like, yeah, let's just fill the other 2, kind of thing, like with some spoonies or something like that.
Yeah.
So we're hoping to get spread the kits out a little bit better this year than last year, and get people results faster.
Yep.
Very cool. Brian, you got anything else? Jeff, you got anything else?
I want to hear this undercover shooting of the farm mallards story.
The wife undercover mission they want to hear.
Yeah.
Their famous Dr.
story.
Phil, we got his name over here. He can't step on the property.
It's actually
how'd that go down?
No. So this was early on, when I was like, the only way to really test whether this other mallard thingy is game farm is to go get game farm mallards, and then and to get we needed another source. So we were catching and banding Mexican ducks here in El Paso, and thankfully, I guess at that time, and now it's like super thankful, there was a bunch of khaki Campbells there, so we caught them and we bled them, and so they were like our alternative park duck, because maybe it was like park duck that was like the problem. And so to get those game farms, I knew we had to go shoot them, and I knew where this was sort of going, and it could be a problem. And so more or less, I asked my wife at the time, and I was like, "You want to go?" And she went. And more or less the whole time I was like, okay, I'm at 20. And I was like, I'll just go to 30. And so shot 30 mallards and brought them back. But the interesting thing is during that time, right? So she shot 30, but they released 100. And it wasn't because she technically missed. 70 of them just went straight east and west. And it's the ocean and like wildlife refuge. So they're going into those areas.
Oh, wow.
At the same exact time, right? You might be all set up, right? Like morning flight, you're like, I'm going to do an all-day sit. And you got these mallards coming in at between 10 and 2. That are probably just feral mallards that you're shooting.
That's crazy.
You have anything else?
Yeah, it's not. So that sort of worked out. And then, but now, since then, we have birds from Kentucky, Louisiana, Illinois, California, and 2 other places in South Carolina. And so, and in every single instance, they're the exact same genetics, and they're the exact same thing that's in Europe, except for 2 things. In Europe, the way they do it is they basically just have them open pen. They want wild birds coming in, so you've got wild in them. So they're usually something like 80-90% game farm, 10 or so % wild, because they've got this open policy. And there's one breeder in South Carolina that told me a story of his grandfather or great grandfather, that he would catch wild birds and bring them in. I guess that would be in the 70s or 80s. And I was like, oh, all right. And we did the genetics, and I was like, oh, holy crap, they're all 20% wild. so he really did do that. And then I was like, is that not a Migratory Bird Treaty Act violation?
Yeah.
Anyways.
You have anything else you wanted to add, Phil? Anything we missed, or anything you want to promote?
No. So we're still taking, like I said, we're still taking applications. The more applications we have, the more folks interested in it, the longer this thing will continue, and I hope it continues in forever. This Duck DNA program. So everybody listening, if they can go on to www.duckdna.com, there's a whole bunch of apply now. Just put in your stuff, and you'll get entered to win. Again, the kit is completely free. If you get it, it comes to you. You sample. You put back the self-addressed envelope into the self-addressed envelope, and USPS sticker on there, and you just send it back to us, and then we do our job, and you'll get your results, assuming all's is well. And so, again, if folks are listening out there and they're interested in the more donations that DU gets, the more of these kits that we can get into people's hands. As a donation, you get X number of kits. So please, contact your DU representative to do that. And
do they have a special?
Just generally tell your DU rep that you love this program and you want to see it continue.
Do they have like a special fund directly for the Duck DNA? Like you can say, I want to donate to Duck DNA through DU.
Yeah.
Nice. Cool.
Yeah. So that could be its own thing. So that your money is going towards this program. And I know the people that are listening and see it. So you'll get a little kit. So we revised them. It'll look like that. So everything is nice and new.
Perfect.
You open it up, and all your instructions are right there with your 3 little vials and your ethanol wipes and everything. And underneath will be the Duck DNA sticker for you and the USPS prepaid postal service tag on there. You just put it right back here and you ship it right back.
Perfect.
And hopefully in a month you'll get your results.
Yeah.
Right on.
Cool. Well, thanks for taking the time, Phil. I really appreciate it. Always fun catching up with you. Are you coming out to California at all this year to hunt?
Actually, I'm hoping to.
Oh, all right.
Yeah,
well, if you do, you got open invite, so let me know.
Thank you.
Yeah,
I appreciate that. Yeah, I'm hoping to come out there to give a talk at Davis, and then hopefully get onto property.
Yeah, sounds good.
Thank you so much. We appreciate you.
All right, thanks Phil.
Yeah, thank you guys.
Thank you for tuning in this episode of Save It for the Blind podcast. You can listen on Spotify, Apple Podcast, or anywhere podcasts are found.